All right. Well, thanks for inviting me to speak. I'm Laurie Barge from NASA JPL, and I'm going to talk about our recent study about the biosignature thresholds for astrobiology on ocean worlds. Or really what this talk is about is how origin of life science, which is one of my main research interests, can be used in our search for life elsewhere. Oops. So before, in case I don't get to the end, I just wanted to acknowledge our lab group. So this is the JPL Origins and Habitability Lab. We study the origin of life, how it thrives on planets, and how it can be detected. And here are some of the amazing students and postdocs who have worked on this research and whose work that I will be showing. And our funding comes from JPL, NASA, and the NSF. So basically, what I want to get across is that when we look for life on other worlds, sometimes astrobiology is misconstrued as just searching for life, but actually it involves a lot of different other fields. And one of the main ones is origin of life and prebiotic chemistry. And it is really important as a person who studies origin of life. I've found this to be quite essential, really, when we're talking about missions and ways we can look for life on other planets. So this is a rough timeline, very rough, very subjective, of the origin of life, going from the formation of Earth. And then after the formation of Earth, there is some abiotic organic chemistry going on in the geological environment. And then at some point that escalates to be more complex, and we call it prebiotic chemistry. And prebiotic chemistry really just means that in retrospect, we know that it resulted in the origin of life. But if you don't know that it results in that, then maybe it's just called very complex abiotic chemistry. And then there's the origin of life, which is an event which is very subjective and not well understood. And then at some point, there is a last universal common ancestor, or LUCA. And this is a phylogenetically derived life that is not necessarily one organism, but is basically the last common ancestor genetically of all life on Earth. And then at some point after LUCA, we have actual evidence of life in the rock record, which is very controversial, and I'm not going to discuss that here. But then it's important to note as well that before LUCA, but after the origin of life, there is still pre-LUCA genes and enzymes. So there is actually biological properties that predate LUCA, but that are post-origin of life. And so kind of zooming in a bit on what origin of life really is, just kind of going in here, it's often looked at as an event on a timeline, but it's not really an event. It's more of a process. And so instead of looking like this, it might have looked something like this. And so really what we would have had is in the environment, some organic chemistry is going on in the presence of minerals and metals and other geological components, getting more complex. At some point, there's an origin of life, whatever we call that. But after the origin of life, this is now a biological process as opposed to an abiotic process. And then you have the pre-LUCA genes and then LUCA itself. And so there's a lot of research in this field, but to kind of summarize one timeline for how this might have occurred. In the beginning, there's mineral organic chemistry. And so you have minerals and ions and metals reacting with organics to produce some small molecules like peptides or nucleotide oligomers. And then at some point, those get more complex, and you have some simple genes. So these would be nucleotide oligomers, like small pieces of RNA. They could be functional even, they could encapsulate metal ions, and peptides as well could do this, but the whole system would be dependent on geochemistry. And then at some point you have very complex molecules, things like RNA that can, really fold or catalyze. You have peptide chains that are long enough to actually function, maybe proteins. It's still abiotic though, because it's not the origin of life. And then at some point we have this metabolism becoming encoded in DNA. So DNA codes RNA, codes protein, which is how life works today. And then you get to these pre-LUCA systems, which are still, genes and enzymes, but they predate the last universal common ancestor. And so the reason I bring this up is because we talk about biosignatures a lot in life detection, but it's important to note that everything pre-origin of life is abiotic by definition, right? And so therefore, some biosignature properties were also present in the prebiotic world. So for example, cellularity is often talked about in a mission context, like looking for membranes or cells, but cellularity was present before life. And there's lots of research showing formation of vesicles and so forth with organic reactions occurring inside them. Metabolism certainly predates life, and this is a very straightforward thing to accomplish some metabolic reactions using minerals and metals in a geological context. There's complex and functional organics, things like peptides, proteins, genes, nucleotide oligomers, those things. When we're talking about the RNA world, for example, that's what that is complex organics that existed prior to having life on Earth. And then, like I said, genes and informational molecules. So looking for, nucleotide oligomers or informational molecules in a mission context, well, those also existed prior to the origin of life to some extent. And so how do we actually then differentiate life from non-life, given that you could have prebiotic chemistry? Maybe not on Earth today, but on other worlds, you might. And so this is our recent study, and this is a figure kind of summarizing what we propose. And this is a figure showing a lot of prebiotic chemistry. So A through E are all things that have been shown in the lab. So these are lab experiments in the literature that have been accomplished abiotically. And then F is actual life. So these include things like simple prebiotic molecules like a carboxylic acid or a nucleotide, biologically relevant molecules like ATP, ADP, lipids, organic polymers like RNA or peptides, functional molecules like iron sulfur clusters, say nested in a peptide, protocells with or without other organics inside them, and then life. And so what we wonder is, if you detected a molecule on another world using some organic detection instrument, what is the threshold above which you could say a molecule that's more complex than this is more likely to have been made by life as opposed to non-life? And so what we argue is that threshold, the biosignature threshold, actually is not a constant, but that it would change depending on what world you're talking about. So consider a world like Earth, where there's life everywhere, and you find a sample and you find some organics in that sample. So this shows Earth. This is the biotic world. And on Earth, there are abiotic organics being formed still. So you can still find some small molecules not made by life. But at this point on Earth, because of life has dominated nearly all surface and subsurface environments, if you find things like genes or a peptide or an enzyme or a membrane, it's almost certainly made by life on Earth. And so the biosignature threshold for a biotic world like Earth is pretty low because most complex organics at this point have to do with life. But now imagine that you could send a mission or an instrument back in time, say to Earth 4 billion years ago, before the origin of life, but while prebiotic chemistry was still highly active and maybe just prior to the origin of life. You might detect things like a peptide or an enzyme, but it would not be biological yet. And it might be quite confusing given the types of organic detections that we are developing for missions. And so this prebiotic world, which could be like early Earth, but also perhaps, say, Enceladus today, we don't really know. So a prebiotic world, the biosignature threshold would be much higher because all of this stuff could be made prebiotically, potentially, because you don't know what the history of life is on that planet. And so the conclusion here is basically that the biosignature threshold varies depending on the world of interest. And when we look for life on other planets, we have to really think about what that means for the different worlds that we are studying. So this has to do also with the history of life on the planet. And so here's a study from 2015 showing some different possible histories of life on a planet. It goes from abiotic settings over here to biotic settings. Some of them are Earth-like, some of them are not. And the black lines just represent kind of the abiotic chemical evolutionary path. And so just to quickly go through this, there's 5 kind of positions noted on this figure. They represent different types of things that might be encountered on planets. And so number 1 here is a bottleneck. This is where if you don't do this condition or this reaction, you cannot proceed to the origin of life. And so even though it's not life, this would be an important detection target for a mission because it could tell you if the planet is able to sustain life or not. There's number 2 here is if you had a non-Earth-like beginning, but it ends up in an Earth-like biosphere. This would be like if we looked for Earth-like life on other planets, which is one possibility. There's number 3 here, where an Earth-like origin of life leads to a non-Earth-like biosphere, which is pretty likely. You've got number 4, where there might be a rapid diversification of prebiotic chemistry, so different prebiotic systems existing in the same planet, but only one of them actually becomes the origin of life. And then finally, number 5 here is, what if you had multiple origins of life on the same planet? What if you had multiple roots of the tree of life? It's possible, right? It just doesn't exist on Earth that we know of, but it is possible. And so thinking of all these alternatives, you can see how the biosignature threshold might vary on different worlds, but also in the same world through time. And so it's worth considering as we proceed with our life detection efforts, what would each of these scenarios look like to your organic detection instrument? What would it look like if you found, you know, a peptide, but it wasn't biological, or a piece of RNA and it wasn't biological? And so finally, just to wrap up this thinking about ocean worlds in particular, Enceladus, where there's been detected organics, and those organics could have been made abiotically, but there's also a lot of interest in looking for life on Enceladus. And so I wonder sometimes, you know, could Enceladus be prebiotically active even today? It's quite possible. And it's not a guarantee that just because a planet has prebiotic chemistry that it will result in a biosphere. And so we have to be prepared to detect signs of prebiotic chemistry as well as biology. And you cannot define that biosignature threshold using Earth-like samples or Earth-like life, because on Earth, we don't have good examples of prebiotic chemistry existing out in nature because life outcompetes it. So the questions for ocean worlds would include, if you take Earth-like prebiotic reactions, how would they be different under Enceladus conditions, for example? What parts of the literature and the origin of life are doable under Enceladus conditions? Which parts are not? What other prebiotic or biotic histories are possible for Enceladus? Might be different than Earth. It might be similar to Earth in some ways. And then what would be, finally, the biosignature threshold for Enceladus or any other planet that we want to look at? So where would you place that boundary where if you detected a molecule more complex than this, that you could maybe safely say it's from life? In order to place that threshold, you have to know something about the prebiotic chemical possibilities for that world. And so that is how Origin of Life can help us with life detection. And I think these two fields should have a much stronger connection. And that's it. So I'm happy to take any questions, I guess, after this or after Peter.
Thanks, Laurie, for a great intro to my talk. So I'm going to give this is an overview presentation on the work that's going on at JPL. I'll tell you the synopsis because it's so highly focused. I'm going to just tell you about the how. I mean, I think in this forum we know lots about the where and the when and the why and all that kind of stuff, but I'm going to really focus on how and the technologies, specific technologies. If you do want the sort of the highest level encapsulation of the philosophy here, I would point you to this short, concise, well-written white paper. Okay, and so today I'm going to tell you about this OWL suite. This is this JPL project. It's a chemistry instrument and a microscope. I'm going to really focus on the chemistry instrument part, but I will tell you that one of the main things that I want you to take home, high-level points, that to address the goals that all of us are interested in and the stuff that Laurie was just talking about, it really needs to do liquid-based separation science. That's something that may not be obvious, and it's a really high-level point. And okay, this is all funded by JPL, and I just want to say none of the work that I'm talking about here uses flight-like mass spectrometers as part of this project, but we are now funded, and we have been funded in the past, to work with Will Brinkerhoff, who is here as well as part of the Emily project. And there we're interfacing the stuff that you see here with Goddard, flight mass spectrometry, and also gas phase separation science. So this is a big team. I'm representing the work of this huge team. Here's like our very first field trip for the pandemic. We're trying out early prototypes. And then just this past summer, this is a photograph of the team at Mono Lake, where after we invented and built these prototypes, we tested them out there. So I think here's just a sort of overview. I'm going to be really light on this, but by the end of this, I also hope you can appreciate how the stuff that I'm telling you about here, it does represent like the most capable integrated suite. And that conclusion that I sort of boldly put out here is you know, it matches the conclusion of all the different studies that you've all been participating in and published and whatnot. So the goal is, you know, we have listened to we've been participating in the community's, you know, description of this and trying to figure out what to do and how, you know, what targets. And this suite is intended to do this maximum coverage of all these lower rungs in the ladder of life, but also at the highest possible sensitivity as well. Okay, so let me start talking about the chemistry part, and this may seem obvious, may not, but you know, if you really want to do this search for life at the molecular level, you got to be able to recognize the really complicated molecules and also simpler ones and to recognize patterns in simpler ones. And so how do you do that? The inescapable conclusion of this, if you start with like a whiteboard, is you need to do liquid. You have to handle liquids. You got to do separation science in one form or another, and you have to do mass spectrometry. You have to kind of do all three of those things. So that informs what we did. So this whole thing, just to, again, to clarify what it means, like looking for patterns in collections of organics. You know, this is what, like Laurie was saying, if you get some prebiotic chemistry and want it to differentiate, you really need to start looking at molecular properties, and you need to be able to plot like the abundance of something against this property. So the canonical example we all talk about is this Lovelock's first description of how long chain organics would be formed in the two different scenarios. So there's a fatty acid biosignatures shown again here on the right, and then the other one, of course, is amino acids that we've all talked lots about them. And then finally, to put it, the chemistry part in bigger context here, although I'm not going to talk about gas phase separations here, you know, there is, you want to use the right type of separation science for the right targets. So one thing that's not really highlighted in the lateral life and all these things is that all these organic molecules have, you know, quite different chemical properties, and that you can't just ignore that if you want to measure them, and we do want to measure them. So if you take and you put together separation science using liquids and gases, and you connect them to the same mass spectrometer, you do the best possible job in looking for life. And that's what the, you know, Will's the PI of the Emily project, and we're working on that with him now. So searching for cells, again, this is work that Chris Linden-Smith and Jay Nadeau and others have done as part of this project. I'm not going to really go into this, but yes, obviously we all agree that looking for cells would be like the smoking gun of finding life. If you take the OWLS project and you just do a very, very simplistic high level, you know, science trace of it, it breaks down like this, you know, so we got organic detection is A, inorganic, and then cells. And then the interesting thing here, and again, there's nothing new here. You guys have seen this restated and shuffled the words and blah, 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 many, many times. But at the bottom line, you can see there's a variety of different instrument subsystems in OWLS that address these different measurements, starting from using the chemical analyzer uses lasers to do the amino acid measurements. Then for A2, it's a mass spectrometer, so it's catch-all for all the different organics you can dissolve and ionize. And then there's ways we can do inorganic measurements as well, using CE. And we can also do it using like a MECA-like instrument. And then there's the measurements of cells that Chris and others have done. And so to bring it all together, if all that was sort of gobbledygook to you, but you worked on any of these Enceladus mission proposals, like, to be clear, you know, so this whole project started before these things were published, but, you know, this shouldn't be any surprise that this is the core of the Orbilander life detection suite, all the stuff that I'm talking about. So SMS is separation mass spectrometry. ESA, that's electrochemical sensor arrays, like the MECA instrument, micro CE LIF, that's the laser mode, and then the microscope. So we worked on these things. I'm not going to tell you about the mass spectrometry development. That would be the top line that I haven't put in here. So again, Goddard and other places working on these high resolution flight-like mass spectrometers, and the nanopore is just, for us, it was a bridge too far. We didn't attempt to work on the nanopore for many of the reasons actually written about in these reports. So what are these oceans and Elvis? A little bit more detail. So we did this project with in mind that we knew that it would be perhaps unlikely that there would be a spacecraft that could fly all this stuff, but we wanted to make it so that you could pick and choose. So, you know, so the idea is we made all these interconnected blocks that then can be selected from depending upon your mission of interest. So you start, and then obviously you have to have a spacecraft to collect sample for you, and then it goes, we have this extractor module, and then we have a fluidic processing. This is like a distribution hub that can move liquids from place to place, and then there's a chemical sensing module. That's like the MECA instrument on Mars. So I mean, I'd advocate that right away, even that core structure there, there's like all kinds of things you learn just from that. But But then what you can do is from the fluidic module, you can go and send material either to a microscope or the chemical analyzer. And you can see there's a line from the extractor right to the microscope system as well. And that would be a way to determine if you had cells and they were busted apart or not. And then you can see the chemistry instrument and the microscope, then they have sub-instruments inside them. And again, I'm really going to focus on the one on the right. And you see that the electrospray ionization has to go into a mass spectrometer. And that's the one I'm telling you. For missions would come from Goddard. And then for this project, it was really important because there's so much data that we would actually have some kind of machine learning to prioritize the data to send back. I'll say something of general interest here that is important and new. We could talk about this for a long time, but the extractor, this is how we can heat and pressurize, and we can, if there were cells there, we could bust them apart and we could create these smaller molecules that you could analyze with the chemistry instrument. We've published a bunch of papers on that. So that's a very interesting feature. If you wanted to be agnostic about the form that the organics took in your sample. So the chemistry instrument here, it's called Oceans. We received an award for redefining the state of the art in this field. So if you're not a specialist in this, this is new and important stuff to people who aren't interested in astrobiology. But as I said earlier, to do separation science, the simplest way to do separation science with a liquid on emission is this technique of electrophoresis, where you just use voltages to do so. This is something that's commonly used here on Earth, but it's never been flown. And it works because different things move at different speeds when they're dissolved in these fluids. And so we have a bunch of different modes. You can see here's the type of data that you see. So the laser induced, the thing on the left, that's for amino acids. In the middle, this shows, just a calibration of sending things into a mass spectrometer. And on the far right, you see what the conductivity detection looks like. And you can see there the peaks go up and down because there you're basically just detecting any charge of anything inside a solution. Of course, the thing I didn't say yet, which motivates this really, is that if it weren't for the fact that this is many orders of magnitude more sensitive than other techniques, it perhaps wouldn't be worth the effort. But if you want to do detection at the limits of detection that that the Orbit report and other studies indicate, you really have to do this liquid-based stuff. So you can see converted into units, most people appreciate it's, the most sensitive thing is 10 parts per trillion for amino acids, but we have other sensitivities for other things as well. The microscope, again, this is Chris Linden-Smith and others. I'll just briefly say, the big thing is here you can make these measurements over a very large field of view, so you don't have to scan really slowly through to search for things, which is a big feature. And then the autonomous software works on both. You can see on the lower right, it actually sorts through microscope data and looks for things that are swimming and can identify them from the bulk flow. And then also mass spectrometry data sets are really large, so we can also scan through mass spectrometry data and try to find peaks as well. So here's where I've spoken for like 10 minutes or so. I'll be very brief. Here's like what the stuff kind of looked like. The big boxes, we're making obviously smaller versions, but I will say the big thing, we used a flight-like thing. We actually operated this entire suite like you would operate a mission, using flight-like computers and commanding and everything like that, which is a big deal. Here's some showing the extractor. Here's what a bunch of the hardware looks like. Again, I'm sure you may not be super interested, but this is a liquid handling instrument, and we have all kinds of flight qualifiable pieces that we can use to do all this work. Here you can see more of the hardware, how it all kind of goes together, and then we can spray into a variety of different mass spectrometers. Here's what the microscope looks like. And now I'm almost at a time here, but I'll just say, yeah, when you sort of put it all together at the field site, it's many times larger than it has to be, but that's what it looks like. And when we went and collected samples from Mono Lake, of course, we found a big wide range of biosignatures, from amino acids to organics to phosphate, malonate, carboxylic acids. We characterized the provenance sort of the water, and then we found a bunch of organisms. So I'm not going to go into details of that again, because I'm just trying to focus on the how of all this. And but let's suffice to say that we built and we invented new stuff for this and validated it in a real mission-like scenario. And that's a big path. It's a milestone on our path to flight. And I can't help but say this, like this whole team worked super hard during the pandemic, and it was a great group. And I'm really proud of everybody for all the work they did. Thanks for your interest. And then we published so many, like lots of papers, and you can find these, you can find lots of information on the internet if you want, or you can just write me and ask. And that's my 12 or so minutes. I'll stop there.